to introduce.
I was just going to start the recording because I had paused it. Okay, are we ready to go then?
I think we are.
Okay, great. I think, hi everyone. I'm Christine Wagoner from the Cure GM1 Foundation, and my daughter Iris also has juvenile GM1 gangliosidosis. And I'm really excited to be with you today to learn more about the phase 3 Azafaros clinical trial. And this webinar will be covering the study, the Navigate study, and the GM1, GM2 portion of the trial. This is a learning process. Every clinical trial is different. We have enabled the Q&A, but we disabled, actually, I see a chat. Maybe that's only for presenters, but we're asking you to use the Q&A rather, and chat has been disabled for this webinar to protect people's privacy. So I'm also pleased that Kathy Flynn from NTSAD and Dr. Caroline Hastings are here, and Aidan Gill is here to answer questions from Azafaros as well. So I'll hand it over to Kathy.
Sure. Thank you, Christine. I certainly echo your words of welcome. Good afternoon, everyone, and thank you all for joining us to learn more about Azafaros' Navigate program. As Christine said, it's a groundbreaking phase 3 clinical trial testing nizubaglistat in children with late infantile and juvenile forms of GM1, GM2 gangliosidosis and Niemann-Pick type C. At NTSAD, where families are certainly the center of everything that we do, we're very excited about the promise that lies ahead. Today, you're going to hear in-depth details about the Navigate trial, learn about the eligibility criteria, and what participation entails. And we really hope that this webinar offers clear and helpful information. And if you or your loved one meet the criteria and you are interested in learning more, we encourage you to discuss the trial with your healthcare provider. And as always, Becky and Deanna, NTSAD's amazing family services team, are always available to support you. So please reach out if you have questions. And I'm sure if members of the Cure GM1 Foundation have questions, they can certainly reach out as well to Christine or anyone on the Cure GM1 Foundation team. We thank you for being with us and for your connection to our rare disease community. So at this time, we're going to continue on, and it's my pleasure to turn over the webinar to Dr. Caroline Hastings. Dr. Hastings is a hematologist oncologist and neuro-oncologist who cares for children with blood diseases, including blood cancers. She also cares for adults with lysosomal storage disorders, and her research in rare diseases includes projects on relapsed leukemia, brain tumors, and Niemann-Pick type C disease. She works on clinical trials for drugs that show promise for treating NPC and serves on related advisory boards. And in her spare time, Dr. Hastings enjoys gardening and animals. Dr. Hastings?
Thank you. I'm going to go ahead and share my slide deck at this time. Oops, what happened to it? Okay. Let me see. I'm not sure why it's not showing up here. Are you able to see that yet?
Not yet.
Nope.
All right, let's see. If I can't get this up for some reason, Christina might just have you pull it up.
Oh, sure. Okay. Here we go.
Great, thank you. So today, on behalf of Azafaros, I'm going to introduce to you the new phase 3 clinical trial called the Navigate study. Go ahead. And next slide, please. And so just as you know, these slides were created by Azafaros for me to share with you today. And I will be one of the principal investigators in the site here in California, one of the US sites designated. So today on our agenda, we're going to talk about the compound isavaglostat, talk about the mode of action as well as the formulation, review prior clinical data from the Rainbow phase 2 study, and then specifically talk about the study design as well as the criteria for inclusion and exclusion on the Navigate study. Next slide. So this is there's a lot going on this slide, and this is meant to show us kind of all at once what we think the actual mechanism of action is for the drug. It does appear to be somewhat unique and have a dual mode of action. It is known to reduce the activity of 2 different enzymes. And in the middle, there's this capsule here, and GCS and NLGase, and these are the 2 enzymes. GCS is glucosylceramide synthase, and NLGase is the non-lysosomal glucosylceramidase. So these 2 is thought to have an impact on. So in the middle here, we have 3 swimming pools, and you're probably wondering what this means. So this is meant to be a metaphor of a functioning lysosome. A lysosome is an important cellular element that is responsible for helping to ingest and then transport cholesterol and sphingolipids throughout the cell to get them to the areas it needs to be in. So on the left side, we see a nice swimming pool with clear blue water. This is felt to be a normal functioning lysosome. And below it is a cartoon depiction of what happens in the cell. Unesterified cholesterol as well as other sphingolipids are ingested into the cell. They are processed through what's called the endosome and then the lysosome. And the lysosome then helps transport these very needed sphingolipids and cholesterol to other cellular components, including the Golgi complex, the endoplasmic reticulum, and also to the membrane. And it gets it also going outside of the cell to areas that it needs to be in. In the middle diagram, we see, well, in this particular case, MPC, but this could also represent GM1. And again, these very important lipids are ingested. They go into the lysosomes, but because of the dysfunctional enzymes, they are not fully processed or transported in the way that they need to be, and we get backup of these contents. And the backup not only prevents the cell from getting these critical elements to other parts of the cell for survival, but can also do inherent damage to the cell. When we treat the lysosome, it starts to clear up. And in fact, we're able to remove the blockades and be able to then process these lipids normally. So down below, you can see for glucosylceramide synthase that there's an early blockage that the isavaglostat overcomes, and then you don't have the accumulation and backup of all of these substrates to the right that then impact the function of the cell. And it's also hypothesized that by also affecting the second enzyme, the NLGase, that this will also have a beneficial impact on the well-being of the cell and potentially anti-inflammatory benefits. Next slide. So we know that in the prior studies in the phase 1 and in the Rainbow phase 2 study, the drug was administered in capsules that needed to be swallowed whole. They were small, but still did require that intact capsule. In the Navigate study, I think this is going to be a lot simpler for many of our patients that may have difficulties with swallowing. These are dispersible tablets. So they start to disintegrate into the mouth, and you can feel like kind of the grains in them, and then you can swallow it with your saliva or by just taking a drink of water. It also can easily be put into an NG or a G tube. Next slide. So the prior study, the Rainbow study, evaluated safety, tolerability, as well as pharmacokinetics and pharmacodynamics of this compound. So the pharmacokinetics refer to the concentration of the drug that you can measure, and the pharmacodynamics look at what the impact is on other substrates, for example. So the main study was 12 weeks long. Patients were randomized 1 to 1 to 1. So that means 30% receive placebo, so the capsule they received did not have drug. 30% or 33% received a high dose and 33% a low dose. And a total of 13 participants were in the study, 7 with GM2 and 6 with NPC. Now, at this time, GM1 patients were not included because there just didn't have enough results as of yet from the pilot study and preclinical data. But there is more information now that is going to allow them to participate in the next study. Of note, after the 12-week study, during which times safety and clinical assessments were performed, then patients were able to go into an extension study and have continued access to the drug. The patients that were on placebo were then randomized to the high dose or low dose because we just didn't really know what was better, but they got access to the drug at this time and continued for the next year. And there were a total of 11 patients that participated in this aspect of the study. The next slide, please. So the study was performed in Brazil, and as mentioned, included NPC and GM2 patients. And it was not specifically powered, with not nearly enough patients or length of time to look at what the clinical impact or the efficacy was. However, the pharmacokinetics and pharmacodynamics did give a lot of good information as to what the best dose would be to trial in a phase 3. So the 11 participants that went on to the expanded aspect of the study, at least 11, I should say, received 9 months of treatment, and others potentially more. Patients on the study were over 12 years of age at the time. Next. So looking at some of the safety effects, it really appeared to look very safe, and as such has been deemed to have a positive safety profile. The most frequent adverse event, or AE, as shown here, was actually skin dryness, which can be very much alleviated with creams. And it occurred in both the high and the low doses. But when we look at this middle slide, when we ask principal investigators to tell us if they think that any of the side effects, whether it be skin dryness, headaches, or others, do they think it's related to the study drug, for example, then they were to acknowledge that. And you can see that maybe there are a lot of adverse events that are not study related or not drug related. And that's because you have to report every medical condition during the course of a study, even if you get a viral illness. As far as the severity, the majority of side effects were mild and some were moderate, and none were severe, leading to the need to discontinue. Next. So one of the questions that was asked as a primary objective with respect to safety was, is there an impact on seizures? Certainly we know that patients can develop seizures, patients with GM2 as well as NPC. There were only 3 patients with GM2 that came on at the time that had a seizure frequency of already averaging about 1 a day. And it didn't appear that there was any significant impact on this, but we have to look at the fact that there's only 3 patients, It certainly did not increase seizure frequency or cause that kind of central nervous system irritability. There was one patient who had a change in their seizure medications, and maybe there was a slight increase in seizures after that, but you have to consider that drug change as well. So here there's no significant and certainly no harmful concern at this time. Next slide. So as well, what was very important was looking at some biochemical changes or pharmacodynamics. And one of the endpoints was looking at reducing glucosylceramide to see if indeed the enzyme was being impacted, because we suspect that the drug inhibits it, therefore there should be a change. And looking at the levels in the blood, for patients with receiving the high dose of the drug, which is the dark blue, there was an 88% reduction, which is quite significant. And for patients that received the low dose, there appeared to be a 64% reduction. Now, there was also a mild reduction in placebo. We can't really explain that, but it's certainly qualitatively very different than what was experienced with patients on the drug. There was no data that looked at the impact of or the change in the NLGAs at this time, no CSF data. Next slide, please. So also, there are a number of biomarkers that we're aware of in these diseases, specifically those that are related to those that have neurocognitive change. For example, neurofilament light chain, abbreviated as NFL, PPCS, previously known as lyso 509, and also the lyso GM2 and GM2. But when we look at, again, this is color-coded with the high dose, dark blue, the low dose, green, and the placebo, kind of a turquoise. There are some reductions, for example, that you can see in these patients with lyso GM2. There's a little more variability in some, and the PPCS, really not much change. But what's very difficult to determine here is how significant this is with very few patients. So we can't really make a statement at this time that there's a significant change between the patients that received the drug and those that did not. But going forward in a much larger study, we should be able to make that analysis. Next. So one of the exploratory objectives, which is something that was, not the study is not powered for it, but we wanted to collect the data on, was looking at the SARA test, which is an ataxia clinical tool that looks at gross motor, fine motor, speech, for example. And doing these clinical assessments on patients, ideally at baseline and then at 12 months. Here we have some data that shows some encouragement. Again, not enough patients, and not all patients, as I understand it, had data collected at baseline. But we could see in the green here that some patients improve in diseases that you don't really expect that to happen. And as well, we did see some patients worsen, and there were some patients that stabilized. So again, this as an exploratory objective, this was really just trying to get a sense of if there's any possibility for some change here, but really not statistically significant. But it does look encouraging at this time, again, as we would not expect improvement, and we would be happy with stabilization as well over a period of a year. Next slide. So overall, the Rainbow study really showed an excellent safety profile, and very importantly, there were no GI adverse effects, no diarrhea, bloating, pain, etc., that we know can occur with miglustat in many patients. It appears to be well tolerated. It's an oral formulation, It does have a confirmed impact on the enzyme, the GCS enzyme that was identified in the mechanism of action. An optimal dose was identified based on these preliminary data. And we do know that, of course, not all the patients received what we consider the optimal dose. They received the lower dose, but there were still some patients, even in that dose range, that had encouraging clinical outcome. And again, as mentioned, we'll need more data on disease biomarkers. Next. So now to discuss the current, soon to open Navigate phase 3 trial. So phase 3 trial is designed to be much larger, enroll more patients, tends to be longer than phase 1 and phase 2 trials. And in addition to looking at safety and some of the parameters with pharmacokinetics as well as the biomarkers, pharmacodynamics, it's also powered to look at clinical outcomes. There will be an umbrella protocol here that's developed, an identical protocol, but 2 independent studies that will run in parallel. One that will enroll patients with NPC, and the other that will enroll patients with GM1 or GM2. And most clinicians that take care of one take care of the other. So we imagine that at the sites that they'll be open, patients will be able to go on to either of these protocols. And it just makes it simpler for administration and monitoring to have one protocol that's the same. However, these sub-studies will be run and analyzed separately, and one will not necessarily impact the other. Next slide, please. This is what the study design looks like. So patients with GM1 or GM2 will be screened, and we'll be looking at the entry criteria and exclusion criteria, as well as their general well-being. There are then baseline studies that are done. Now, the screening and the baseline can be done together to make it efficient. Then at this time, after they come on registered on the study, there's a randomization and it's done 2 to 1. For every 2 patients that go on the drug, 1 will be on placebo. This will be double blinded, so we know that neither the investigator or the patient and family will know which drug they've been randomized to receive. There will be clinical assessments done in 1 month, and then at month 3, and then every 3 months through the end of the study, which is 18 months in duration. Patients can continue after that to go on to open label and continue to have periodic clinical assessments. At that time as well, patients that were on the placebo will go into open label and receive the drug. And presumably they'll continue on open label until all patients have completed the study and there's a period of time of analysis and a submission to regulators. And the next slide. It is anticipated that 75 patients will be enrolled. Again, mentioned this is a placebo-controlled study. And for approximately 50 patients that receive the drug, 25 will receive placebo. So the dose is going to be adjusted to body weight. So even though it may look like there are 3 different doses, patients will get the same approximate dose when you consider their body size. So it'll be 3 milligrams, 6 milligrams, or 9 milligrams. Next slide, please. Patients and families must be able to provide written informed consent, which can be either by the participant, parent, or legal guardian. They must have a confirmed diagnosis of GM1 gangliosidosis or Tay-Sachs, Sandhoff, or GM2 AB variant disease. That's based on confirmation from enzymatic tests, identification of 2 pathogenic variants, or 1 pathogenic variant plus what's called a VU subvariant of unknown significance, but it's a mutation basically in the same chromosome, in the same location, but it hasn't previously been identified as being that disease. And it's just because it may just be a newer mutation. So it can be a variant, a VUS, and confirmation with low enzyme activity. Patients must be at least 4 years of age, and there's no upper limit of age. Onset of neurologic symptoms between 1 and 10 years of age. And the SARA study will be done, or SARA assessment will be done at baseline and calculated. And a very low score is minimal symptoms, and high scores are very symptomatic. But patients must have a score of at least 3, but be less than 30. And the reason for this is that the trial does not want to enter on very severely affected patients who are not likely to have benefit, nor will they want patients that are progressing so slowly that we won't be able to determine if indeed they're having a clinical change over the 18-month period. Next slide, please. So these are some of the exclusion criteria. So as I mentioned, they must their well-being must be good. So there should not be any medical conditions such as severe liver or kidney disease that can interfere with any interpretation of the results or performing the study assessments. Patients must be able to participate in all of the study assessments. A body weight under 10 kilos is not allowed, and that's because of the formulation of the drug and the lower dose. But again, we don't anticipate patients being this small under when they're 4 years of age and older. If they have seizures, they must be on stable seizure medications and not be in the process of changing either the dose or the drugs. So once they're stable, they can come on to the study. They can't be receiving another investigational study, participating in a clinical trial with an experimental drug at the time of or in less than 3 months. But if they were on it 4 months ago, then they've had a good washout period and they can come on to the trial. Also, use of miglustat is prohibited. Next slide for more discussion about that. So miglustat has a very similar mechanism of action to nisobaglustat, so we don't want patients to be on both drugs because it would be confusing to understand which drug may be having the impact. Patients, however, can be considered for enrollment if they're on a subtherapeutic dose of miglustat for 6 months or longer, and their clinician or investigator considers that the treatment has really not been effective. Patients can be on some newly approved drugs. So this is more specific to NPC, Ecnirsa and Miplyffa, though I know that there are some patients certainly looking at Ecnirsa, but the indication now is an NPC. It's not approved in all countries, only approved in the US at this time. But if there are approved drugs that are considered standard of care, they can be on these drugs if they've been on them at least 3 months. And supplemental medications, including Tanganil, cannabinoids, and curcumin, amongst others, are allowed as long as the PI is aware and can report on that. And again, stable doses for at least 3 months before the study and during study. Next. So at this time, there are 4 centers that are participating in an initial submission in the US, in California, Minnesota, Texas, and Virginia, potentially more US centers in 2026. And this is an international trial, so there will be patients enrolled in a number of clinical sites in various countries, including Latin America and Europe and Australia. And the plan at this time is to start before the end of 2025, hopefully by the fall or maybe even late summer. Next slide, please. So I'm happy to take questions. And just for clarity, Christine and Kathleen, are all the questions in the Q&A that are visible